The nuclear behavior in tapetal cell. A common feature of tapetum, amoeboid as well as secretory, is that the total DNA content of the tissue increases enormously through a meiotic prophase. This is achieved by one or more of the following ways. First is the multinucleated condition. Such a situation arises when during mitosis, nuclear division, karyokinesis is not accompanied by wall formation. Cytokinesis. This is a very common feature of tapetal cell. There may be 1, 2, 3 or rarely 4 such mitosis in a tapetal cell. According to the cell, will have 2, 4, 8 or 16 nuclei. However, if there is a nuclear fusion or some mitosis fail, cell with the nuclei number outside the expected series may appear. In the parietal tapetum, the nuclear division rarely synchronous, whereas in periplasmodium type, they are highly synchronous. Second is the endomitosis. This is a type of mitosis in which the chromosome duplicates and the chromatid separation takes place within the intact nuclear membrane without the formation of spindle. This results in a large polyploid nucleus. In the uninucleated tapetum of cucurbita pepo, nuclei of 4 size corresponding to their degree of polydy 2n, 4n, 8n and 16n are formed as a result of endomitosis. Formation of restitution nuclei. This also results in polyploidy nuclei. Mitosis goes on normally up to the early anaphase stage, but the two set of chromosomes finally get incorporated into a common nuclear membrane to form a restitution nucleus. Fourth one is polygyny. This refers to an increase in the number of chromata per chromosome. The mode of DNA increase does not alter the number of chromosomes per nucleus. Next, the function of tapetum. The tapetum appear to play a significant role in the development of pollen. First, during the pollen formation, during meiosis, it is assumed to be simply transporting the nutrient to the inside of the anther locule, since this is the only canal through which material can reach the meiocyte. At this stage, the nutrients are not derived from the tapetum. Mehen and Lane 1969 have demonstrated that in Tradescantia bracteata, the plasmodial cytoplasm derived from the tapetum has a callous activity. Shortly before callose degeneration starts, the cytoplasm of the tapetal cell shows certain vesicles which are probably associated with callous activity. Third, morphologically and cytological study on the anther development in male fertile and male sterile lines have shown that the formation of non-viable pollen in the sterile line is associated with non-functional and faulty development of tapetum. Franklin et al. In 1969 reported that the microspore mother cell do not show callose wall after prophase first. Estimation of callase activity in fertile and sterile line revealed that this abnormality is due to faulty timing of callose appearance in the cytoplasmic male sterile plant, whereas in the fertile plant callase appear only at the late meiosis and the tetrad stage. In the sterile plant, strong callase activity is detected during the prophase first. Based on the point 2, it may be concluded that the precocious release of callase by the tapetum is probably responsible for the cytoplasmic male sterility in Petunia. Fourth one is, during post-meiotic period, tapetum play an important role in pollen wall formation. Its contribution of sporopollenin through UBIS bodies. Fifth point is, most conspicuous example of the transfer of material from the pollen tapetum to pollen is of the pollen kit substance and trypsin, whereas the former appear to be the complex mixture of hydrophobic liquid and carotenoid. The latter seems to be a complex mixture of hydrophilic substance including protein. Pollen wall 6 point. Pollen wall contain protein derived from the gametophyte as well as the tapetal cell. The protein of the later origin are presented in the space and cavity of the exine. These proteins are rapidly released when the pollen grain are moistened and are responsible for the common hay fever and pollen caused injury allergy. Another biological significance of this protein is the recognition of compatible pistil. If a pollen land on an incompatible stigma, these proteins include the formation of callus plug in the stigma or style and block the growth of the pollen tube. Similarly, callus plug are formed in the tapetum, exercised at the onset of dissolution of its applied to the incompatible stigma, confirming that the tapetal origin of the substance responsible for the rejection reaction by Hasselhoff and Harrison et al. 1974. Thank you.